Good day, I'm Melinda Kliegman, the Public Impact Director at the Innovative Genomics Institute, which is a collaboration between the University of California, Berkeley and the University of California, San Francisco. Founded in 2014 by Jennifer Doudna, with the goal of using genome engineering to solve some of humanity's greatest problems. As part of our strategy, we focus on CRISPR applications for human health and continuing to develop innovative new genomics tools. Relevant to our discussion today, a significant portion of our work is also focused on climate change, and it is a core part of our mission to advance public understanding of the benefits of genome engineering and to ensure that any benefits of the technology are advanced in an equitable and ethical way. So we've already heard from researchers who use genome editing in their labs and who have painted a picture of the current approaches to use genome editing to address climate change. But as a breeder, you may ask, what happens after one develops a genome edited trait in their lab and tested in the greenhouse? What would be the next steps? And so the answer to this is that the time and cost does depend partially on where you are located. So at a very simplified level, researchers will identify and develop new improved approaches and tools that they can use to identify new traits. And these traits are sources of productivity, stress tolerance, or helps with climate adaptation. So these traits are then introduced into the breeding pipeline where they are integrated into high-performing varieties. And then here on phase 4, things can really change depending on whether the product is categorized as a transgenic or a GM product or GMOs in a specific country. The researcher may have requirements around field testing the traits. If the product is deemed to be a GM product, they may need to put together a significant regulatory package of data that includes a battery of tests for product safety. And then they will need to take that to the country's biotechnology regulator for review, and that review can sometimes take years. If it is deemed that the product is not regulated or similar to a conventionally bred product, which is the case for some genome edited products, then the product would not necessarily need to go through that process. And I'll talk a little bit more about that later. But from there, any product, conventionally bred or GM, would need to be scaled up and distributed via the appropriate pathways for whatever particular system you're in. So where exactly is that line drawn between what's considered to be a genetically modified or GM product or living modified organism or a genome edited product? So globally, regulators have taken a very practical approach that is informed by the 35 years of experience that they have with transgenic or GMO plants. Most regulators globally did not want to have to issue new laws, although some are doing so, for example, in India. New laws can be really difficult and take time to move forward. So instead, they've opted to interpret their existing laws to determine whether genome edited products fell within the existing definitions of a GMO, and then determine from there whether those products can be excluded from their oversight and treated as a conventionally bred product. And most of the language in existing laws was drafted in the late 80s or early 90s. It makes a reference to recombinant DNA technology and a novel combination of genes as a trigger for regulation. So with some types of genome edits, like single point mutations, deletions, and introduction of DNA sequences from sexually compatible relatives, a lot of regulators have deemed that this is not a novel combination of genetic material. And so some countries had said that these products would not be regulated by the rules meant to provide oversight for biotechnology, although all food is still subject to rules around food safety, which is similar to what conventionally bred products also have to go through. So there's also another practical reason why regulators have chosen this approach. If something could have occurred naturally or by conventional breeding, it's really impossible to tell which products are genome edited in order to require or ask for more data. So regulators are also thinking about enforcement and also just the general point of regulations, which is for safety. And if you can't tell the difference, then there's really no point in regulating. So as you can see from this map, most of the countries in the Americas have taken this approach. And several countries in Asia and Africa are considering similar approaches. Japan has taken a bit more of a conservative approach where they're saying if any DNA is introduced, so even if it's from a sexually compatible relative, for instance, they're considering that to be regulated. The one outlier was the EU, where a court case determined that genome edited products fell into their definition as a GMO, as was written in law. But earlier this year, they have said that the law as written is not fit for purpose. And they're likely going to go about updating that language, hopefully with more flexible language to allow them to better adapt to new technologies in the future. Some countries haven't said anything yet, and a lot of these countries are treating genome edited products as they are GM until they can establish their laws. So just to recap, if you are considered to have a transgenic product, you may require multi-location field trials and regulated as a GMO, and need to submit a significant regulatory package. But if the product falls into a category of an excluded genome edited product, it may just be treated as conventionally bred and can move forward in the pipeline, potentially saving the developer a lot of time and a lot of money. And in some cases, it could mean the product getting out at all. There are examples where, due to public perception and regulatory and political roadblocks, some products have just never been put on the market. And I think the most famous example is the transgenic humanitarian golden rice to prevent childhood blindness. So how big is this difference in cost and time? So industry experts estimate that it can take about 13 to 14 years to commercialize a GM product, with about 5 years of that time taken up with collecting the data for the regulatory dossier and waiting for regulatory approvals. And they estimate a much reduced cost and time for genome edited products with about 5 years and $10 million. So particularly if we look at a crop like wheat, for which there's currently no GM varieties on the market, genetic diversity for disease resistance has been lost over time through bottlenecks from polyploidization and domestication. So genome editing could play a role here, perhaps, in bringing in disease resistance genes from wild relatives or developing varieties that can adapt to a range of climatic conditions. And since wheat does not generate a lot of farm revenue compared to maize, it has not really been a target for genetic modification, since presumably industry can't really recover the cost associated with the research investment. But if we have a reduced cost and shorter times, then perhaps we might see some more public researchers or small companies looking into different solutions. And is this what we're seeing in practice? And the short answer is yes. So in 2015, Argentinian regulators pioneered the model for how to regulate new breeding techniques. They do require a case-by-case approach. So developers need to come in to regulators to have their products reviewed, and products that don't have a novel combination of genetic material is released via existing health and safety rules for conventionally bred products. So that was now 6 years ago. So we have some early data. In 2020, the key regulators in that country published a paper summarizing what they found. And this provides an early example of the impact of regulations on innovation. So right now on the screen, this is the type of developers that have submitted applications for transgenic GMO products. The first approval was made in Argentina in 1996 of a herbicide-resistant soybean. And you can see from this graph that most submissions, 90% of them, were made by foreign multinational companies. So if we compare this to genome-edited applications by developers, we see a wider range of distribution of who have submitted applications. We actually have more developers, so 13 different institutions for genome-edited products, compared to just 10 institutions for the GM product developers. And this is a comparison of 23 years of products submitted for the GMOs versus just a 3-year period evaluated for the genome-edited products. And down below on the second panel, we have the types of products that have been submitted. And if you look at the difference between GMOs or GM products and genome-edited products, there's a much wider variety of genome-edited products submitted. And here again, fewer applications, fewer total overall number of applications in a shorter time frame, but still a much wider variety in just those 3 years. And so what this highlights to me is what people have been saying for a long time, but not with a lot of concrete evidence. It shows that the cost of regulation can really shrink the number of players that are able to operate. And even for large multinational companies, they are limited to what they develop because they can only focus on crops that can be the most profitable since the regulatory burden is very high. And so overly onerous regulations can actually end up having the opposite effect. I think a lot of calls by civil society organizations for increased regulations on GM products is aimed explicitly at limiting the activity of large multinational companies, but the outcome is that it's too expensive or difficult for all but the largest and most well-funded players working on a tiny fraction of traits and crops. And so that leads to more consolidation, it leads to monocultures. And by lowering the regulatory barrier, you can see a widening field of companies and local and public research institutions. Because you have a wider range of institutions, you're going to have a wider range of products because people have different interests. And some of these new products might be consumer facing, related to sustainability or climate change, instead of solely focused on increasing yields and profitability. So this is exciting to me. I think it portends a really interesting future as the landscape continues to evolve. And I personally think one of the potentially biggest beneficiaries could be smallholder farmers. Another thing that came out of the Argentinian study is that there's a big increase in the quantity of applications over time for genome edited products. It's rising very quickly. And so if we put this all together, the products are easier and faster to develop, potentially cheaper. This just means they're more accessible. And this is all particularly useful in LMICs. So the CGIAR, which is a top class research center located in LMICs, I think they operate in over 100 countries globally, have said that the average age of a variety in a farmer fields in the countries in which they operate is 12 to 25 years, compared to 3 to 5 years in the US for corn, soybean, and for barley in Western Europe. So using these old varieties really leaves farmers at risk for evolving pests and diseases in a changing climate, and it deprives them of the progress of breeding. And so I think what is powerful about genome editing is that it could lead to more responsive breeding because of the shorter turnover times. And so breeders can provide a steady stream of new varieties to respond to changing climatic conditions. But to make this a reality for LMICs, it's going to be really important for us to learn from the past and to empower local breeders to develop and use the technology themselves. So, scientists in the CG system, local national agricultural research institutes are really important because they understand the local context and know what the local farmers, consumers, and environmental priorities are. And one of the big criticisms of GM technology is that it was Western developed. And so I think here it's important that this not solely be a Western technology, but be homegrown. So the last thing I want to talk about is public perception. We can't really ignore it. I think it's a big part of why GM products have not been widely adopted in LMICs, and a part of the negative feedback loop of why there are high regulatory requirements and consumer rejection. So this is a big concern that we as scientists need to appreciate and try to address where possible before products hit the market. So to give you an example of some data, Yang and Hobbs in 2020 published a study on the perception of genetically modified and genome edited foods. They asked people to rate from 1 to 6 whether they thought foods produced with a particular breeding technique was natural, ethical, and safe to eat. For this graph on the y-axis, we have the rating scale with 1 being not at all safe, natural or ethical, and 6 being completely. And the colors are darkest blue for crossbred plants, which received the highest ratings across the board. Light blue for mutagenesis, which I think it's funny because it's considered a conventional breeding technique, but it's rated quite lowly. The horizontal hashtags are GM or transgenics, and the hashtags at an angle are genome edits, which received somewhat intermediary scores here. And when we look at other similar studies published to date, we see a similar distribution. So these studies have measured people's attitudes, perceptions, or willingness to buy products and at what cost. And I've grossly oversimplified the studies here so I can compare them roughly. But you can see conventional breeding techniques generally receive a highest positive mark with the 2+ signs. Genome edited products are usually somewhere in the middle in regards to perception, or there's mixed perception among different countries surveyed. And then the 2 negative signs indicate that consumers had a negative association with the breeding technique, or at least the most negative compared to the other options with which they were presented. And so I think overall it shows that consumers are still a little bit more open to genome edited products. And then with the question around mutagenesis, it really shows you how framing and kind of preconceptions about a particular word can have an effect on how individuals think about a product. This second paper that looked at mutagenesis is from Australia, and they showed that when consumers found out that they were already eating products of mutagenesis and it was considered a conventional breeding technique, that their perception of it improved. So what can you as a scientist do about all of this? So in the US, the public has a fairly high perception of scientists, and this has been relatively stable since the 1970s. And then in addition, a lot of people are getting their news from social media for better or worse. So you as a scientist have a direct platform to provide information about your work to your friends and family who will likely trust your opinion. And I know I'm nervous about doing this, but then I'm reminded by Instagram influencer dietitians in my social circle who have no medical or scientific training, freely giving out advice to their followers about why kale is bad for you and why you should never eat it. And I realized, okay, maybe I do know a little bit more about this. And so you shouldn't be self-conscious about engaging because you likely do have some training. And if this is not the space, if social media is not the space in which you want to engage, because it can sometimes be a little hostile, you can still make a difference through structured outreach programs at your university or local science centers or museums. I like to say if you're going to use the tools in the lab, then you should be invested in conveying about its safety and usefulness. And most importantly, why the use of technologies can be aligned with societal values. and how these new tools can really help us reach our goals around sustainability and climate change. So thank you so much for your time and attention.